Welcome to our first lab here. Today we're going to have an orientation lab. We want to talk about some of the terms we have to know so that we can just have a common vocabulary when we refer to parts of the body or structures that we know what we're talking about. Everybody's on the same page. We're also going to talk about the microscope, what we can see with the microscope, and why this is so fundamental to our appreciation of how the body works and what's going on here. So at the end of this lab, what you're going to do is that you're going to take a lab quiz. The lab quiz is online. You take the lab quiz. That's worth 10 points, and score some. Add some points to your score here. So make sure you take that lab quiz sometime this week, because after the week is over, then class moves on to the next lab, and that quiz will no longer be available, and you'll be out 10 points there. Okay, so let's get started and see what we're up to here. So with the lab tasks for each week, we can just look at the lab, look at the material. When you want to study, the PowerPoint slides are on Canvas there, our Canvas class page, and you can just go through there, look at the questions, figure out what you have to know and how you have to answer the questions. Take the lab quiz and get your points here. So we need to know what we call the standard anatomical position for humans here. And so every single physiologist is going to subscribe to this. And the standard anatomical position means that the patient is standing and arms are at the sides. The palms are facing frontward. Now, clearly, the palms, you can rotate your hands so the palms are facing in the other direction, but in standard anatomical position, the palms are facing forward, the thumbs are facing toward the side, or what we call laterally. We have things toward the head, which is going to be superior. Things toward the feet are going to be inferior because we're standing here. When we say right and left, then it's the patient's right. So this is the right side of the patient because this is the patient's right. This is the left side. So when you're working on a patient here and they say cut off the right one, then you're talking about the patient's right, not your right, not my life, not my right, but the patient's right. So you never make a mistake here. We've got an easy one here. I've got the midline here, which is just right smack dab down the middle here. I get proximal, which is here on the limbs here at the top. I'm moving in the distal direction. If I move in the other direction, go from the hand toward the shoulder, I'm moving in the proximal direction here. Same thing applies to the legs here. Here, if I'm moving toward the body along the leg here, that's proximal. If I'm moving toward the extremity, toward the feet, that's going in the distal direction here. I have to the front here, when we're standing, that's going to be anterior. The back is going to be posterior. Now, there are two terms here. Anterior is often referred to as ventral, and posterior is referred to as dorsal. In principle, the field that uses ventral and dorsal is neurology. Basically, all the other fields, they're using anterior and posterior, but we have to make friends with our friends from neurology. So remember, anterior and ventral are synonymous. It's the same thing. It's just that neurology uses ventral, and they're not going to change. So remember that distinction here. Here are the planes of section here. If I make a section here, the frontal section, I'm cutting the patient into an anterior and a posterior section here. Then that's going to be called a frontal section, also called the coronal plane. If I go at 90 degrees here, sagittal goes right down the midline here. This is going to be called the sagittal plane. If I cut my patient up like a sausage here, I can cut it here, 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 just like you'd cut a sausage. That's going to be the transverse plane here. Now, just as an aside here, if you're dealing with a horizontal animal, now anterior and posterior, they're towards the head and towards the tail here of the animal. That sort of makes sense. And now dorsal is going to be up in this direction. Here we would consider this superior here, but here this is going to be in the dorsal, and that's going to be the ventral. And so these terms, dorsal and ventral, anterior and posterior, they kind of really originated with in the history of it from horizontal animals. When humans stood upright, then they had to make a little transposition. These are the ones we want to worry about. Where it's frontal, cut the patient into an anterior and a posterior, which is going to be sagittal. I cut the patient into a right and a left half. Transverse plane, I'm cutting the patient up just like I'd cut a sausage here, a number of slices here. So here, if I look at the brain, here's going to be a frontal section because I'm going right through the midline here. I can make that more lateral or, more midline here. I can cut a number of sections here. Here is going to be the coronal section. I can just cut this back and forth here. So all those things are going to be same terminology wherever you look at the body. And so we know what we're talking about here. Here are one of the things about the standard anatomical position is that the going towards the head is always in the superior direction here. So here I have two positions here. Prone means the patient is lying on the stomach here. Okay, so the patient is in the prone position here. And now superior, if I say I'm working on the patient, move in the superior direction, you're still going toward the head because this is superior in the standard anatomical position. If I say move in the inferior direction, it's toward the feet because here. And now posterior is the back, anterior is the front. So this is the prone position. Patient is lying on his stomach here. Here's going to be the supine position. The supine position, here the patient, she's lying on her back, still superior toward the head, inferior still toward the feet here. And now anterior is this side, upward, because this is the anterior side. Patient is flipped over, posterior is on the other side. So all these terms, they are relative to the patient standing in the standard anatomical position here. Let's look at some of the internal structures here. And it turns out that when you look inside the body here, all of the organs, they're all surrounded by a membrane. And the reason for this is that friction is really the enemy of the body here. So all of your organs, the heart, lung, all these things, they're all surrounded by a membrane that is secreting the slippery stuff so that every time you move, that slippery stuff reduces friction here. And friction is the enemy of the body. If two tissues rub together, not only does that make the body have to work harder, but now these cells are going to become damaged, and it's a terribly serious condition when we have a breakdown of one of these serous membranes. Normally, okay, I get the slippery stuff, and so remember, the real job of the serous membrane is to reduce friction. It also tends to hold the organs where they're supposed to be, and it turns out that, one of the really surprising things when you cut open, an animal or a patient, you look inside, it looks exactly like the textbook because there is a right answer in where these organs have to be. And if they're going to serve the body and contribute to the survival of the individual. If these things are in the wrong place, body's not working correctly. And when these things are in the wrong place, typically the surgeon will go in there, put it back in the right place, patient recovers, and okay, now he's on his way. But if it's in the wrong place, patient feels terrible. It can be a life-threatening condition here, so membranes are important here. Let's look at our microscope here. So here's just a standard microscope, and we have a number of magnifications that I can look at the microscope here. And I've got two sets of lenses here. The objective here, I have a bunch of lenses in here, and I have the eyepieces in here. And the total magnification, how much bigger of what I'm looking at is it than reality here? And so I take the magnification of the eyepiece, and it'll be written on here, say 10x or something, and what is multiplied by the objective magnification might be 4x, it might be 10x, it might be 40x, and just multiply those two numbers together. So if the objective, the eye part, was 4 times, and this lens here was 10, just 4 times 10, my total magnification is 40x. Whatever I see when I look through the microscope is 40 times larger than it is in reality. Here, 10 times 10, it's 100, 40 times 10 is going to be 400. And this is what kind of magnification we're looking at when we use the microscope. And it's important to realize that, microscopes are very useful. It's enormously helpful to help us to understand how the body works here. Before microscopes, people had no idea that cells existed because, most of them are too small to see with the unaided eye. And the ones that you can see with the aided eye, there's no reason to think that that it's a single cell. Who knew what a cell was here? So here's a diagram. Here's the eyepiece. Here's the objective. And your basic microscope. And this is a light microscope, meaning that, okay, I have a light bulb here. I can shine the light through here, and I can see. And these are really useful. I get a lot of magnification. I can see how cells work. I can tell when cells are healthy. I can tell where they're unhealthy. And as we saw in the previous slide, I can go up to about 1,000 times the actual size there. So these are very powerful, useful tools in our field of study here. And what can you see with the microscope? When you look at human blood here, I can see that there are a bunch of cells. And before the microscope, these cells, they're too small. You can't see that they're cells in blood. It just looks like a bunch of red stuff here. And so as soon as the microscope came about, people began to be aware that blood was actually made out of these individual components called cells, and that there was not one cell. There were several cell types. There's little round one called red blood cells. There's other classes of cells, and these are called white blood cells. And there are a number of cell types here that without the microscope, you're completely unaware of. You look at pond water, you can see all these organisms like this. Here's one that's tetrahymena, much too small to see without a microscope. As soon as you get the microscope, you see all kinds of things that are swimming around in the ponds and streams, and good for you here. Pine pollen, you can see pine pollen, it's sort of granular, gives you hay fever, you can see it, but you can't really see any fine structure to it. And with the pine pollen, you can see not only is it tiny, I can barely see it here, but it also has these two little wings here to enable it to catch wind currents so that it can distribute itself far and wide and contribute to the survival of its species here. Now here's a picture here that shows the typical diameter of a human hair here, and this is the size of a red blood cell here. So it's tiny, compared to the diameter of your human hair. And much smaller than a red blood cell are going to be bacteria. And, much smaller are going to be some other kinds of bacteria some are rod-shaped, some are rounded. And when I look at viruses are much, much smaller than bacteria or cells we find in the body here. They're so small that I cannot see viruses with a light microscope. It is just beyond the power of them. And for that, I need other kinds of microscopes. And we're going to talk about the electron microscope in just a few slides here. But let's look at what we can see here. So when I look at cells in the human body here, one of the first types I want to look at is skeletal muscle cells. So skeletal muscle cells, any muscle that you can voluntarily move. I can move my finger, I can move my legs, I can do whatever I want. Anything I can voluntarily move. This is skeletal muscle. It is under my conscious control. I will my body to move a part, and I can see it. And when I look at it under the microscope, it turns out that these are very long cells. This muscle cell goes from the entire length of the leg here, the entire length of the arm here. So these are just giant, giant cells. And when I look at them with the microscope, I see that these cells, not only are they enormously long here, that they've got all these stripes in that they call striations here. And these striations, they're actually protein molecules within the muscle cell that are lined up so precisely that I can see them at the light microscope level. An individual protein molecule is much, much too small. Can't see it with the individual protein molecule. But if I line them all up in a row and make that row thick enough here, now I can see them. And this is, very easy to see here. So name the cell that I'm looking at, skeletal muscle, and this is voluntary, meaning that I can control its movement just by the will, just by thinking about it here. The next kind of muscle is cardiac muscle. So it turns out if I look at the heart here and I take a sample from that muscle, it looks different. It's got the stripes here, but it's not now organized in these nice, neat cylinders like we saw before. They're kind of X-shaped, they're Y-shaped, there's spaces around them here, and they're not long. They're kind of short. From here to here, that's one cell from here. So what the purpose of this here, it's not made to, move an arm or move a leg here. It's made to squish blood out of a chamber. So it kind of forms like a little netting here, almost like a fishnet here. And so these cells are X-shaped or they're Y-shaped here. They're all fused together here. And when they contract, then what they're going to do is they're going to contract this whole structure here into a, they're going to squish that chamber, squish the blood out, and enable you to pump the blood and go about your business here. Now, this is involuntary, meaning that you cannot control your heartbeat. And okay, the good thing is that, well, when you fall asleep, you don't have to worry about your heart stopping because it's all under an automated system here. And you can't speed it up, you can't slow it down. It's under its own guidance here. How much oxygen does the body need? If it needs more oxygen, heart rate increases. If it doesn't need any more, heart rate is going to slow down, try to conserve energy here. So, very useful, involuntary cardiac muscle. Now, let's look at the difference one more time. When I look here, then I can see that skeletal muscle, nice cylinders here, nice and parallel here. When I look at my cardiac muscle, now I can see that X or Y shape, lots of space in between here. Third muscle type is smooth muscle, and smooth muscle's got no striations, none of those stripes at all here. And they're kind of spindle shaped here. They've got this pointed at both ends here, and that's how they're going to appear to us in the histological section here. And what these guys do, these are involuntary. They surround the hollow organs of the body. Hollow organs, they're like your stomach, they're like your intestines, they're like, a lot of the tubules that connect various organs in the body. Anything that's hollow is going to be run by this smooth muscle, because the job of the smooth muscle, when they contract, they make the opening smaller. They say, let's stop stuff from going through. You want to have more to here? These things relax, the hole gets bigger, I can pass things faster here. And that's the job of the smooth muscle. But again, involuntary. The body's logic is that, well, one of the things the smooth muscle is running is going to be digestion. As far as the body's concerned, digestion is much too important to let you, make a decision on things that you're going to screw it up here. So everything is controlled by a central part of the nervous system. And okay, that's good. Every time I eat, I don't have to, think about, well, how am I going to digest it? That is all an automated system run by these smooth muscles. So these guys, they can contract with great force, and they can hold those contractions for hours and hours at a time. So perfectly adapted to our digestive process here. Now, to summarize, here we started off with skeletal muscle. Everything's nice and parallel here. I can voluntarily control this. I will my arm to flex, send the signal down here, muscle contracts, arm moves here. Cardiac muscle, it's a net, no voluntary control here. And it's made to squish blood out of this chamber. Very efficient. Doesn't fatigue. From the about 3 or 4 weeks of, your embryonic life, the heart starts contracting. You start moving things around here because I just have to have that blood supply to keep those cells alive. Without the heart functioning at the very early stages, the fetus is not going to survive because I can't get enough oxygen to them. So it goes from about 3 or 4 weeks of embryonic development all the way to the day you die. And as soon as you die, of course, you can't exist without the heart here. So that's pretty much going to be the end of you here. Smooth muscle. Now it's going to help me digest food, regulate blood flow throughout the body. Extremely useful. So voluntary, I can control this. Cardiac and smooth muscle, both of these are involuntary. They're under an automated system to make sure that the right decision is made and that all is going to be well in the body here. Let's look at a different cell type. These are adipocytes. So it turns out that there's cells in your body. So this is a cell, and it's got this little, here's the nucleus, it's got this little membrane around it. And right in the middle of the cell is this big vacuole here. This is actually, fat. So it's used here, and it turns out that fat is a really useful way to store an energetic molecule. It is the most efficient way to store energy here. It's going to be in this fat molecules here. So here I have these fat cells here, and when you run out of energy, when you don't have enough to eat, the body comes down here, breaks off some of these fatty acid molecules, metabolizes them, makes ATP, which is going to be energy here, and keeps you going here. When excess energy is there, now it puts that, saves it for a time when I'm going to run out of energy, fills up these fat cells, and that's how the body is preparing for when things go wrong, when I can't get enough to eat here. So here's, if you're familiar with the sport, it's you see this in Japan, sumo wrestling. These guys are big guys, and they're let's be upfront here. They're fat, and so this is part of the sport. That there are those skinny sumo wrestlers because the idea is to try to push your opponent out of the ring here. So the more you weigh, the better off your chances of winning the event are. And one of the really interesting things is when people looked at the physiology of these really, fat sumo wrestlers, their blood pressure is really low. Their cholesterol level circulating is really low. These guys are actually very healthy, and it's not completely understood why these guys can just, eat like it's going out of style every day of their lives and be quite healthy. After they retire from the ring, there they tend to live very very long healthy lives here. So being fat is not as detrimental, or we don't quite let's put it this way, we don't quite understand why being fat in some cases, like in the United States, is so detrimental to your health, whereas these guys, whatever they're doing, doesn't seem to affect them in the least here. Let's look at nerve cells. And nerve cells have one of the most variability of shapes. I've got all kinds of dendritic branches here. I've got all kinds of shapes, and they twist and turn. They go throughout the body. These guys can be extremely long. They can go from one end of the body to another here. And so here's a picture of one kind of cell called the Purkinje cell. This is a pen and ink drawing made by this guy, Santiago Ramon y Cajal, here in 1899. So in 1899, he's got a relatively primitive microscope. Here's a one made from present day, the identical cell here, and they kind of look alike here. In some ways, you can see more detail in the ink drawing here than you can with this fancy dye fill projection that is made in the present day here. All right, other kinds of nerve cells. I've got every kind of variety, and they typically have a very specific shape here. So here they've drawn a single cell here just so we can see what they look like. But when you look at it in the brain, I'm going to have hundreds of thousands, maybe millions of these cells packed from one end to the other. And they all pretty much have the same kind of shape and size here. And this is going to be, when they're assembled, this is going to be how the brain is going to work and how we're going to make our way in the world here. Here's the skeletal system. So here's the typical steak you get at the supermarket. And if I look at the bone on the steak here, it's just bone. It's just this white stuff with this little, darkened area in the center here. But if I look at that white stuff under the microscope, this is what it looks like here. It's quite organized. I've got these little cylinders here. Here's one. Here's another. Here's another here. And at the center of this cylinder, there's a little hollow space. And in the hollow space, I've got blood vessels, I've got nerve cells. So it's highly organized here. And not only is it organized, they're living cells here. Each one of these little black things, these are living cells here. So that if you put some stress here, if you start to lift weights, if you start to learn how to play a sport here, the bone gets thicker. It says, there's some stress here, I don't want to break, I'm going to make this bone thicker here. So it's a living tissue here that's able to become stronger when you stress it. And when you don't stress, this construction work, that's not for me, I think I'm going to go to school, then make that bone smaller. Bone is heavy, so it makes the bone smaller so you have less weight to carry around. And okay, here we are. But what we want to remember in this slide here is this is a picture of compact bone at the microscopic level here. Doesn't look like it when I look at it with the unaided eye, but as soon as I put it under the microscope, clear as day, I can see that they're living cells. I can see these structures that are going to make this bone able to carry out its function here. Here's connective tissue. This is a kind of tissue that's that makes things like the tendon. So if you look at the back of your leg, there's this tendon that goes right down the back of your ankle here, called the calcaneal tendon here. And when I look at that under the microscope, it's all this wavy stuff, these little black things. These are the nuclei of cells that make this connective tissue here. And it's all wavy because when you stress this, when you jump here, then this thing stretches out. So all these waves, they can absorb a lot of shock here, and they can enable you to jump, to absorb shock when you come landing down here. So again, a form of tissue that's perfectly adapted to its function, which is going to be to generate a lot of force that can enable me to jump, to walk, to run, to do whatever I want to here. Now, single cells are usually too small to see without a microscope, but there are exceptions. So if I look at an egg, any eggs, here's a chicken egg, but I could use an ostrich egg or any kind of an egg there. And when I look at that egg, this is a single cell. There's one cell here. The yolk here is going to be some nutrient material that the cell is going to use if that egg is fertilized, and that growing embryo is going to use the energy in the yolk in order to divide, in order to make an offspring here. And so, of course, people have been looking at eggs, since, forever here. But nobody realized that this was an actual cell here because, why would you? It's just an egg. But be aware, whenever you see an egg, it is a cell. I can see it clearly with the unaided eye here. And single cell here. Here's a kind of single-celled organism. So this is big enough that I can clearly see it. It's like a microorganism here. And this is going to be a organism that's called a stentor here. And so let's look at our stentor here. And
the most beloved of tiny organisms is probably the tardigrade. Bumbly, cute, chubby, and six-legged, they look like macro animals, hence their common names, moss piglet and water bear. And look, we love them too. We love them, and we will talk about them on this channel a great deal. But we also have a favorite species that you likely have never heard of. Larger than a tardigrade, though only a single cell, and the fact that you've probably never heard of stentor is sad, because they are beautiful, magnificent, and powerful organisms. So, stentor. It's a genus of single-celled eukaryote. There are 19 known species, but there are probably more than that. Some of them can build mucus houses that they can hang out in and hide inside of. Some carry algae inside of them that produce food for them. And all of them are astoundingly large for a single-celled organism. Some can be as much as 4 millimeters long, big enough to be seen without a microscope. The biggest tardigrades, which have thousands of cells, are 1.5 millimeters long. One thing every stentor has in common is the holdfast organelle, which lets them anchor to a single location. Now, they can swim, but in an undisturbed culture, only a few stentors will be swimming freely. Stentors are actually more dense than water, which means that they sink, so that holdfast organelle lets them save energy once they find a location that has plenty of food and oxygen. But just because they're anchored, that doesn't mean they can't move. Most stentors are also capable of stretching a huge amount. When attached and feeding, a stentor body can reach 5 to 10 times their original length. And that's when it starts to resemble a trumpet, which is where it gets one of its common names, the trumpet animalcule. The biggest of all the stentors, and one of the largest single-celled organisms that exists, is Stentor caeruleus. They can be the length of a rice grain. There are insects in your backyard that are smaller than these massive but common unicellular organisms. They're in freshwater habitats all around the world, and we, here at Journey to the Microcosmos, have a pond where we find them all the time. Surprisingly, they thrive even when it's freezing cold. We have a stentor culture set from Stentor caeruleus collected under 20 centimeters of ice. Possibly the lack of predators during the winter keeps their numbers extra high. Stentor caeruleus is a part of a group called the ciliates. As is true of all ciliates, Stentor caeruleus have hair-like structures called cilia. The beating of the cilia propels these cells in the water when they want to move around, and it also brings food particles, microanimals, and single-celled organisms into the cell mouth, where they are taken inside the cell to be digested. Just like this multicellular rotifer, which is swallowed by the giant Stentor caeruleus and waiting to be digested. We recorded this struggle for a long time and even witnessed the rotifer rupturing the stentor's cell membrane multiple times. Each time, the stentor repaired itself and the rotifer never managed to escape. After 25 minutes, the rotifer ceased its struggle. Just a reminder, rotifers have a simple brain and a simple nervous system. They can feel, I'm not sure about pain, but they can certainly feel stimuli. The striking blue color makes Stentor caeruleus one of the most beautiful species of the genus Stentor. However, Stentor caeruleus are not just big and blue. They also have many abilities you would not expect to find in a unicellular organism, including regeneration, light avoidance, food selection, and reaction to mechanical stimuli. Stentor caeruleus is most famous for its regenerative abilities. If a Stentor caeruleus cell is cut in half, each half will regenerate into a normal-looking cell at half the size of the uncut cell. After healing and reconstructing the missing parts of the cell, these half-sized cells will grow to the normal cell size given time and resources. Even if the cell is cut into 100 pieces, each fragment can eventually become a normal-looking cell. Though for successful regeneration to occur, we need two things. The cut piece must contain at least part of the macronucleus and a piece of the cell membrane. The macronucleus of Stentor caeruleus is visible even under low magnification. It has the look of a beaded necklace, and it extends along the whole cell. This macronucleus is highly polyploid, which means that even a fraction of the macronucleus will contain thousands of copies of the entire genome of Stentor caeruleus. Even a tiny fragment less than 1/100th the size of the Stentor caeruleus can reconstitute itself in this way if both the cell membrane and the macronucleus are present. Portions of stentor lacking either macronucleus or cell membrane only survive for a short time, and the whole regeneration process takes only around 48 hours. And you don't have to take our word for this. Sometimes, when preparing slides, we accidentally cut these massive cells, and the pieces of Stentor caeruleus are left alive in the slide. Within a day, they are almost fully regenerated, and after 2 days, we can't even tell which is the fragment and which are the unharmed cells. Stentor caeruleus can be cannibalistic as well. However, no one has ever managed to record the initial swallowing. This is likely because the light of the microscope disturbs the cells. Here we see a stentor making a go of that, but luckily for the little one, it's a bit too big of a bite. As for that blue color, we know why, but we don't know, why. They contain a blue pigment, stentorin, but we don't know what purpose this beautifully colored chemical serves. We have guesses. It's possible stentorin helps Stentor caeruleus sense light somehow, but there's also evidence that when a predatory single-celled organism touches a Stentor caeruleus, it ejects the pigment like squid ink. In some cases, scientists have observed the predatory organism pulling away. Unfortunately, though we have attempted to record this process for ourselves after many hours of attempts, it still hasn't happened while recording. But remember, this isn't the only species of stentor. Here we have Stentor polymorphus. This species is mostly filled with endosymbiotic algae. These algae live inside of the Stentor polymorphus and produce sugar via photosynthesis and give some of the extra sugar to Stentor. In exchange, Stentor provides protection for the algae. Look how small those algae are. If you are surrounded by thousands of different species of algae eaters, hiding in one of the biggest things around would certainly protect you from getting eaten. Look how big they are. Each dot is a Stentor polymorphous cell. These cells came from an aquatic snail. It had so many Stentor cells on its shell, it was visibly green. Now, we've been talking about these beautiful and peculiar organisms for a long time now, and there is, of course, still more to share. We'll be working hard to keep our stentor cultures alive and healthy, and we haven't even talked about the peculiar way that ciliate's genetic code works and how stentor is the only ciliate that uses the same system for encoding genes into proteins that you and I do. We'll have to leave that for a future video. For now, enjoy these big blue beauties. And hey, thanks for coming on this journey with us. If you want to see more from our master of microscopes, James, follow Jam and Germs on Instagram. And if you want to see more from us, that, my friends, is what the subscribe button is for.
All right, so that's stentor. Got another one, single-celled algae. And so they are around us, these single-celled organisms that can't be seen, but without the microscope, we would have no idea that these were single-celled. They just look like, eggs. Here's the light microscope view of bacteria. And, bacteria, they come in rod shapes, they come in like little, sort of curled ones, they come in spheres. But of the, hundreds and hundreds of thousands of bacterial species here, at the light microscope, they kind of basically all look alike. I can see that, well, some are round and some are rod shaped, but beyond that, I can't see very much just because these things are just too small. I can't look inside them. I can't make any distinction here. So, okay, these are at the small end of the scale on which I need a different kind of microscope if I'm going to understand what is it that bacteria do. What are they up to here? Now, that other device that I can use to look at things that are too small for the light microscope is something called the scanning electron microscope. And with the scanning electron microscope, instead of using rays of light, then I use rays of electrons. So I have this thing that's spraying electrons. The electrons come down here. Now, if you're spraying electrons, how are you going to focus electrons? I can't focus an electron with glass because the electron just goes straight through glass. It doesn't work. And the answer is that when I want to focus an electron beam, I use magnets because electrons have a negative charge here. I can use magnets to change the shape of the beam just like I would do in, light coming through glass here. So I have a series of magnets here that I can vary the field strength here, and I can focus that beam onto here. And when I focus the beam onto here, now that beam reflects the electrons. I can detect that reflection of the electron. And what's so good about the electron microscope is when I look at something with the electron microscope, not only can I see things that are too small to see with the light microscope, but I can see them in 3 dimensions here. So here's bacteria like we saw at the light microscope, and now I can see there's a shape here. There's a front, there's a back. I can see that there's these little, sort of fibers these things are using to stick themselves together with. Now, okay, these things have been false. Somebody came and colored these things because the electron beam doesn't have a color, but still, the example holds. When I look at something with the scanning electron microscope, I see them in 3-dimensional form. And as I'm looking at it, I can move this thing. I can move my frame of reference. I can look all around it. I can turn it from side to side. I can do whatever I want. So very, very useful here. Here's a scanning electron microscope of a simple fly here. And I can see a lot of detail here. So here's a low magnification with the scanner. Here I can see, the veins on the wings here. I can see the eyes. I can get a close-up of the eyes. I can see these little spikes on the eyes. I can see details on the wings. I can see little details on the feet of the fly here. These little fibers here, they're gonna enable the fly to, basically walk on the ceiling here. And so this is how they do that here. Now, the next kind of electron microscope is not the scanner, but something called the transmission electron microscope. And now I'm using a much more higher powered beam here. So the beam is much higher powered here. I'm still using magnets to focus the beam here. And now I can get much higher magnification here. So when I get the higher magnification, this is what bacteria look like. I can see inside the bacterium. I can see there's a cell wall, there's a cell membrane. I can see these particles in there. I can see lots of details that I can't see with the other two types of microscopes that we've looked at here. And now we're sort of on the road to understanding, what's going on inside of the cell. What is enabling this particular cell type to live here and survive in its environment here. I can look at cells, but now with this transmission, I can only look at a part of this. I can't back it up to look at the whole cell because it's just too big. I can't fit it in there. But I can see there are all these other organelles here. There are these little, membrane-covered structures here. These little oblong things, there are little circular things here. And now I can look with the scanner and I can see all these other structures. And now I can figure out, what does a healthy cell look like? And by looking at these structures, I can tell, what is this cell capable of doing here? So again, very useful. I can get the kind of information that I cannot get with light microscope, that I cannot get with the scanning electron microscope. And this is going to be transmission electron microscope here. Here, I can look at pieces of the membrane. I can look at pieces of the endoplasmic reticulum here. And these pictures here in the transmission, they are all two-dimensional. I can't look at 3 dimensions like I can with the scanner here, but I can look at much higher power here. The other thing is that with these electrons, because they're electrons, there is no color. Well, I could go in there, I can color them any color I want, and people do that all the time, just to make the structures stand out a little bit better than they normally would. Now, another thing to realize with these electron beams being focused on tissue is that they're quite high energy. You can't shine an electron beam on cellular tissue and expect that they'll survive. It just completely obliterates it because the energy levels are too high. So whenever you see, particularly in electron micrograph, what you're seeing is not the actual cell. You're seeing a cell that has been sprayed with an atom, typically osmium or sometimes silver, that they coat these things with so that it doesn't destroy. So you coat these things that you turn on the beam, it obliterates all the real cell tissue, but I have the replica here. So I'm actually not looking at the cell, I'm actually looking at a mold of metal ions that are sprayed on this thing in order to make this thing work here. Now, let's look at the microscopy of movement here. So this is one of the great things that we can look at. Not only can I look at things as they are, but with my light microscope, I can actually see how things move here. So let's look at a video here of movement that we can see with the light microscope. All right, so I can see movement here. I can capture them. I can video them. Everything's going great here. Let's look at this other device here, and this is something that it actually is a device that you can put on your cell phone to record. Make your own microscope here. Now, the thing is that if you want to go out and buy a microscope, a lot of people say, I kind of like to buy a microscope. You got to go up to about $300 or $400 before they even start being anything worthwhile here. And so, okay, that's a lot of money here. So it turns out, okay, you got a cell phone. I've got a lot of electronics in there, and this is going to be a device that's going to enable you to use your cell phone as a microscope for about $35 to $40 here. So let's take a look at what this thing is here. All right, so available through Amazon or any basically kids store that you want to go to here. So okay, this brings us to the end of our lab here, and next you're gonna look up lab quiz 1. It's going to be worth 10 points. So what do you have to know about the slide set? So study before you don't just look at this and take it. Make sure you look at the slides and study them. And once you do that, you start the lab quiz there. And what you need to know: the standard anatomical position, know the terms, know the orientation, know which is superior, know which is inferior, that whole thing. Know the planes of section of the body, just like frontal section, transverse section. Know the histological sections in this lab. Know what it's going to be a slide. What is this? Is it skeletal muscle? Is it nerve cell? Is it smooth muscle? Is it cardiac? What is it? Be able to identify that here. Tell me of the muscle types, which ones are voluntary, which ones are involuntary, and what does that mean? What does it mean when muscle is voluntary? What does it mean when muscles are involuntary? Know the different kinds of microscopes: light microscope, scanning electron microscope, transmission electron microscope. What can you see with each type of scope, and what can you not see with each type of scope here? So if you're looking at viruses, you're pretty much looking at transmission electron microscope. If you're looking at insects, well, I can use light microscope, I can use a scanner. But if I want to look a little bit in 3 dimensions, then I'm really looking at the scanning electron. So just know some of that detail, know how to interpret these things. And these are just the basic tools there. Understand how the same thing can look quite different, depending on what kind of microscope you use to look at your sample here. So, okay, good luck to you on your lab quiz. Make sure you take that sometime this week, because after that, quiz goes offline and points are no longer available to you here. If you have any questions or comments, come to email me with what you think, or good, bad, or indifferent, what you liked about this lab. Particularly if something about this lab was just really confusing and you had no idea what was going on, then by all means, don't just sit there and take it. Email me and say, well, explain this to me. Why is this so important here? So, okay, I wish you well, and I will see you next time. Good luck and stay in touch. Keep up.